The monomers, or building blocks of DNA, are nucleotides. Nucleotides consist of three things: a deoxyribose sugar, a phosphate, and a nitrogenous base. There are four different nitrogenous bases: adenine, thymine, guanine, and cytosine. Certain nitrogenous bases attach or fit together through hydrogen bonds. Adenine and thymine are attached together through 2 hydrogen bonds. Guanine and cytosine are attached together through 3 hydrogen bonds. DNA is formed into a double helix shape, with nucleotides on either side of it. The nucleotides run anti-parallel to each other. For example, the phosphate of one nucleotide will be going upward, while the phosphate of a nucleotide on the opposing side will be going downward. Transcription is the DNA-directed synthesis of an RNA molecule. In other words, it is writing DNA in a different genetic code. Transcription begins at the promoter of DNA. Promoters are simply what initiate the process of transcription. RNA polymerase is the main enzyme in transcription. An enzyme is a biological catalyst that speeds up reactions. The RNA polymerase breaks the hydrogen bonds between the nitrogenous bases of the DNA and brings and attaches the RNA nucleotides to the DNA. RNA has the same nitrogenous base pairs, except, unlike DNA, which contains thymine, RNA has uracil. So uracil connects to adenine, not thymine. Transcription ends when the RNA polymerase reaches the termination sequence of the DNA. This is a sequence in the DNA that indicates the end of a gene. The process results in a messenger RNA strand, or an mRNA strand, of a single gene being produced. RNA editing is done between transcription and translation. The purpose of RNA editing is to edit the mRNA created from transcription to ensure the correct protein is made. First, a cap and a tail are added to either side of the mRNA strand. The cap and tail act as a protective barrier so the molecule doesn't break down. Then, the non-coding introns are cut out from the molecule, leaving the remaining exons to be pieced together. The end result is a mature mRNA strand.
Translation is the process of translating the information in the newly created strand of mRNA into a chain of amino acids. Once the mRNA is fully edited, it leaves the nucleus and goes into the cytoplasm, where a ribosome will do the actual translating. Ribosomes are organelles that have 2 large subunits, which are made up of rRNA, or ribosome RNA. When the mRNA reaches the ribosome, it begins to be threaded through the middle of the 2 subunits of the ribosome. As it's being threaded through, the letters of the mRNA are read 3 at a time. Each 3 letters is called a codon. Whenever a codon is read, a tRNA or transfer RNA molecule with corresponding anticodon, the opposite letters of the codon in the mRNA, travels down to the ribosome and leaves behind a certain amino acid. Which amino acid depends on the codon that was read, because each codon codes to a certain amino acid, though some codons all code to the same amino acid. When another codon is read, the process repeats and the previous amino acid is bonded to the new amino acid, the peptide bond. There are certain codons that are called start and stop codons. These tell the ribosome where to start and stop translating the mRNA. Eventually, there are enough amino acids to form a full polypeptide chain. There are 4 stages to a polypeptide chain becoming a protein. Stage 1 is called the primary stage. This is where the amino acids are bonded together and become a chain. There are a number of factors that determine what the eventual protein will be, including the number of amino acids, what amino acids are in the chain, and the sequence of the amino acids. Stage 2, the secondary stage, is the point at which the chain first starts to fold due to attractions between amino acids and the hydrogen bonds connecting them. Chains will either fold to form an alpha helix or a beta pleated sheet. This is what an alpha helix looks like. This is a beta pleated sheet. In the third or tertiary stage, the chain continues to fold so that the hydrophobic amino acids are oriented towards the inside of the chain, and the hydrophilic ones are oriented towards the outside. Hydrophobic means afraid of water, and hydrophilic is the opposite, meaning it doesn't want to interact with water. For some proteins, this is the final stage of folding, and the chain is now a protein. However, some chains have a 4th stage called the quaternary stage, which only occurs if there are 2 or more polypeptide chains involved in the making of the protein. This stage involves folding both of the chains together to form a fully functioning protein. The main point of this is that DNA creates RNA, RNA creates proteins, and proteins create you. So if it weren't for this process and the capabilities of DNA and RNA, you wouldn't even exist because much of your non-water mass is protein. Proteins also serve as enzymes, transporters, supporters, regulators, defenders, communicators, and more, which allow your body to do the basic things that make sure you don't die a painful death. So in the end, if it weren't for DNA, there'd be no RNA. If it weren't for RNA, there'd be no proteins, and if it weren't for proteins, you'd be dead.